Welcome, medical students, to today's lecture on the fascinating topic of eosinophilic esophagitis, O. O is a chronic immune-mediated disorder characterized by eosinophilic infiltration in the esophageal tissue, leading to inflammation and dysfunction. In this comprehensive script, we will explore various aspects of O, including molecular mechanisms, T helper cell differentiation, esophageal barrier dysfunction, predictive factors, unique challenges in elderly patients, genetic predisposition, esophageal remodeling, endoscopic techniques, association with other allergic disorders, microbiome dysbiosis, predictive models for dietary interventions, and novel biomarkers for diagnosis and monitoring. O is primarily driven by the recruitment and activation of eosinophils in the esophageal tissue. We will delve into the specific signaling pathways and molecular interactions involved in this process. Key players include various chemokines, such as eotaxins, and their corresponding receptors on eosinophils. Additionally, we will discuss the role of adhesion molecules, such as intercellular adhesion molecule 1, ICAM1, in facilitating eosinophil migration into the esophageal tissue. T helper cells play a crucial role in orchestrating the inflammatory response in O. We will examine the different subsets of T helper cells, such as Th2 and Th17 cells, and their cytokine profiles. Th2 cells are known to secrete cytokines, such as interleukin, IL-4, IL-5, and IL-13, which contribute to eosinophilic infiltration and tissue remodeling. Meanwhile, Th17 cells produce IL-17, which promotes the recruitment of neutrophils and enhances mucosal inflammation. One of the hallmark features of O is the dysfunction of the esophageal epithelial barrier. We will investigate the molecular mechanisms underlying this dysfunction, focusing on alterations in tight junction proteins, such as claudins and occludins, that regulate paracellular permeability. Furthermore, we will discuss potential therapeutic targets for restoring barrier function, including drugs that modulate inflammation and promote epithelial repair. To guide therapeutic decisions, it is essential to identify factors that can predict disease progression and treatment response in O. We will review various clinical, endoscopic, histologic, and molecular factors that have been proposed as predictive markers. These may include symptom severity, endoscopic findings such as esophageal rings and furrows, histologic eosinophil count, and alterations in gene expression patterns. Understanding the utility of these factors will aid in tailoring personalized treatment strategies for patients. Diagnosing and managing O in elderly patients present unique challenges. We will explore the impact of age-related comorbidities, including cardiovascular diseases, on the clinical presentation and treatment options. Furthermore, we will discuss modifications to treatment approaches, considering potential drug interactions and the presence of polypharmacy in this population. Genetic factors have been implicated in the development of O, with evidence of familial clustering and specific gene variants associated with the disease. We will discuss these genetic factors, such as filigree gene mutations and polymorphisms in genes related to T-cell regulation. The potential implications for genetic testing and counseling will also be explored. Chronic inflammation in O can lead to esophageal remodeling, characterized by fibrosis and alterations in the extracellular matrix. We will delve into the underlying mechanisms, including the activation of fibroblasts and secretion of profibrotic cytokines, such as transforming growth factor beta, TGF-beta. The impact of esophageal fibrosis on esophageal function and potential therapeutic interventions aimed at preventing or reversing fibrosis will be discussed. Endoscopic evaluation plays a crucial role in assessing treatment response and disease monitoring in O. We will review various techniques, including mucosal biopsies and imaging modalities such as endoscopic ultrasound and confocal laser endomicroscopy. The advantages and limitations of each technique will be discussed, along with emerging technologies that hold promise for improving diagnostic accuracy. O shares underlying mechanisms with other allergic conditions, such as asthma, atopic dermatitis, and allergic rhinitis. We will explore the epidemiological and pathophysiological links between O and these comorbidities. Common immunological pathways involving abnormal activation of T helper cells and IgA-mediated immune responses will be discussed. The implications for management and potential shared therapeutic approaches will also be addressed. Investigations into the esophageal and gut microbiome have revealed alterations in patients with O. We will explore the evidence on microbial dysbiosis and its potential role in disease pathogenesis. The therapeutic implications of targeting the microbiome, such as probiotics and fecal microbial transplantation, will be discussed. Furthermore, we'll analyze the impact of diet and potential interactions between the microbiome and dietary interventions. Personalized treatment strategies, such as elimination diets, have shown efficacy in some O patients. We will review existing predictive models and biomarkers used to identify patients who are likely to respond to specific dietary interventions. Clinical outcome measures, genetic markers, and the potential for combining multiple biomarkers to improve prediction accuracy will be discussed. Advancements in biomarker research hold promise for improving the diagnosis and monitoring of O. We will explore the potential of specific molecules, genetic markers, or non-invasive tests for diagnosing and monitoring O. Biomarkers like eotaxin 3, periostin, and specific eosinophil gene transcript levels may aid in diagnosis, while non-invasive tests such as saliva and urine samples may provide alternatives to invasive procedures. Future prospects in biomarker discovery and clinical implementation will also be discussed. To summarize, our study on eosinophilic esophagitis highlights the various intricate factors involved, including inflammation, immune response, genetic and environmental factors, and potential treatment approaches. Understanding these complexities can lead to improved diagnosis and patient care.